Okay, so in the last lecture, which I guess you will watch on your own, we talk about mutations, right? You know, how exome sequencing is very important. It can help us identify important genes for tumorigenesis and cancer progression. And in that lecture, we talk about, you know, like some genes can be drivers. Those mutations really drive or cause the cancer and accelerates the cancers. Whereas other mutations can just be passenger, because a very important hallmark of cancer is genome instability. You might have a driver gene which initially caused the DNA to be mutating more than normal. And as a result, some other gene mutation just happened, right? And then they get passed on to all the daughter cells as the cell is amplifying, is proliferating. And so it's just the byproduct of DNA instability. And so what people found is that in order to really separate the drivers from the passengers is what you ask, if you have to sequence hundreds of individuals, whether a particular mutation happens recurrently in many patients, that recurrent mutation might be the likely driver. For the high throughput driver detection, you know, people have tried different things. You could use mutation, but people also look at expression. You know, if this gene is always higher expressed in cancer compared to normal, that's probably an important oncogene which cause cancer, or tumor suppressor, which normally suppress tumor, but in cancer somehow is lost, right? You can use the expression. You can also use copy number variations. In the early days, before people can use sequencing, they have already started using arrays. For example, SNP arrays, which, you know, pretty much covers all the SNPs in the genome. And based on that, you can see that a chunk of the human genome get amplified. And so early days, people just use copy number. Yeah, you can see in this case, if you compare, say, the tumor with the normal, say the blood, and you look at the copy number, most of the time they are close to zero. Just means this is log chain, log difference, right? So log difference is cancer normal of zero, means there's no difference between the cancer normal. But then you could see, oh, there's a big chunk, all the probes, suddenly the tumor seems to have lost DNA in that region, right? That's a copy number loss. And so in addition to mutation, copy number is also a very important thing. So this is a work done at the Broad Institute and Dana-Farber Cancer Institute. They developed something called a GISTIC score, or a G-score. And they just basically ask, if we profile hundreds or thousands of tumor, can we see, so it's a very simple score. They ask, what is the frequency of the amplification or deletion, and the magnitude or the amplitude of the copy number change. And then they ask, whether this is a very strong or very weak score. So for example, if say this particular mutation is appearing in 10% of the patient, then the frequency would be 10%. But the average amplification, so normally we have 2 copies, but when you amplify, it could be like 3 or 4 or 5 or 6, right? You can ask, you know, if we calculate the kind of the frequency and the average amplification, you can ask whether this is really significant. So they can look at whether some of these amplification are significant by, you know, random permutation. And because they check every gene in the genome, you do need to worry about multiple hypothesis testing. But you can imagine this is one tumor, this is another tumor, this is a third tumor. They all have mutation in different locations. But after you collect hundreds or thousands of those tumors together, you look at this chromosome, you suddenly see this one region where tumors are recurrently having an amplification at pretty high copy number. Then you know that this is a significant aberrant region. Whereas, the significant score can be determined by just, if we were to randomly put these mutations in different locations, what you would see. Okay, so you can see, that's a significant mutation. So if you see something like this region is amplified. Sometimes for copy number, the region can pretty big. In order to figure out which gene might be the one, very often people also look at gene expression. If this region, could have 10 genes, but one gene is really upregulated a lot, that gene might be the real driver gene, whereas other ones, maybe just kind of passenger. And so in the previous lecture, we also talked about this idea of tumor suppressor versus oncogene. So tumor suppressors normally suppress tumor for unchecked growth, continuous growth. There need to be some checks. Whereas oncogenes are the ones that just really it's like the brake and the gas pedal. In order for cancer to happen, very often what you have is a gain of function mutation on the oncogenes or loss of function mutation on the tumor suppressor gene. A lot of these oncogenes very often are genes that were important when we were a little embryo or when we were kind of growing during our puberty, right? Those are supposedly genes that receive external stimulus to say grow, like they get some growth hormone signal, then they start to grow. But in cancer, for some reason, they did not need a growth signal anymore. A mutation happened, then the gas pedal is always on, right? That's one situation. Tumor suppressors are, just to make sure that the cell, at every amplification, you want to check to make sure the cell is okay. If something goes wrong, you should just let the cell die or at least do not allow it to move forward. But then maybe because of the mutation, this check mechanism is gone. So tumor can continue to amplify the DNA. And so based on the mutation patterns, we can see whether something is a tumor suppressor or oncogene. You can also now couple it with gene expression patterns. And of course, really, in order to prove it, you need to do some functional studies. We will tell you functional studies based on like CRISPR screens later on in the module. Interestingly, some genes can be tumor suppressors and oncogenes. So we'll give you some examples in this, okay? So if you look at cBioPortal on one gene, this is cBioPortal, we'll show you later, is a website where a lot of cancer cohort data have been analyzed, and you can just visualize it on the fly, whatever gene you are interested in. So this is one gene called MYC. If you look at the different cohorts, so each of this bar, so each of this in the bottom is a different study. Say this is TCGA lung cancer, this is TCGA bladder cancer, this is TCGA breast cancer, and so on. And when you see this red, you can see this is amplification. So this gene is amplified a lot. And if you look at gene expression, MYC expression is much higher in the tumor compared to the adjacent normal. And if you look at the mutation, there are some mutations, but not as much. I would say not any really strong recurrent mutation on a particular base. So MYC mostly, so these green ones are the mutations, and you can look at this, but you can see they are not dominant for most of the time. You see a whole DNA copy number amplification and higher gene expression. Can you guess whether this is an oncogene or a tumor suppressor? It's an oncogene, yeah. Because it's like a gas pedal. When you have a gain of the copy number and higher gene expression, they drive cancer growth, okay? And this is another one. This one, if you look at the gene, there are not so many amplifications. Instead, you see a mutation. Right? See, the red is amplification, green is mutation. And if we look at the mutation patterns out of, I guess, TCGA studies, you can see this one new, amino acid location can be changed to many other different, so one amino acid changed to something else. And it's appearing in 1,395 cases. It's that one place gets suddenly mutated. Can you guess whether this is an oncogene or tumor suppressor? This is an oncogene. This is the exact case where this gene needs a growth signal in order to let the cell proliferate. But because of this mutation now, the gas pedal is on all the time. most of the time when you have a mutation that happens on a very specific location repeatedly, that's a sign it's an oncogene. And because of the mutation, you can call it like a proto-oncogene. The normal form is okay, but the mutated form is oncogene now. Okay? Yeah, so it's that one position which take out the condition is unconditional growth, constitutive gas pedal in some sense. And let's look at another case. In this case, you can see now we in all the different cancer cohorts. Oh, by the way, this y-axis tell you how many percentage of those tumors harbor this particular type of mutations. Yeah, you don't see, see occasionally some amplification, but probably more deletions now. And you also see mutations, right? And on this different mutations, you can see missense mutation means there is an amino acid change. And in-frame mutation means you have an insertion and a deletion of a full amino acid. Whereas truncating mutation means it's a frame shift, and then there's an early stop codon. The gene now is shorter or can be degraded much, much faster. And so you can see there's actually a lot of truncating mutation which make the gene shorter in terms of the protein level. It's making a smaller, shorter protein. Can you guess whether this is an oncogene or tumor suppressor? It's a tumor suppressor, exactly. Because first of all, you see copy number loss, right? And if you look at the cancer types, you will see that very often this gene has lower expression as well. So each can give you some indication that, like it's a tumor suppressor. And also, when you have mutations that are kind of spread out, you don't see one place that's super high. Even each of these, like in some sense, each of these mutations make the protein work a little less. The break is not working very well. Or truncating mutation, the protein is like shorter. It just doesn't work as well. And so that's an indication this is something which prevents the tumor for growth, and somehow it gets lost. Okay. This one, you can see, it's interesting. If we look at the mutation pattern, we don't see a lot of loss. It's mostly mutation. And you look at where the mutation happened, you see a lot of truncating mutation here. There's a loss, but then you also see 2 or 3 places, which happens in hundreds of places. Can you guess what happened there? Is P53 a oncogene or tumor suppressor? It's both. Yeah, so if it's a truncating mutation, you will have a protein loss. That's so it turns out the wild type P53, when it's normal, it prevents the tumor growth. Actually, it's important for DNA damage repair. If something is wrong with your DNA, P53 is there to help with fixing those errors or sensing or fixing the errors. So if you have a truncating mutation, you lost the protein, those, you will kind of lose a repair mechanism, then patient will more likely to develop cancer. Interestingly, if the patient have a mutation on those locations, not only the patient will have more DNA damage repair, somehow the mutated form in these locations, they become drivers they become oncogenes in this case, or potential drivers. So people have tried this in some cell lines. If you create those mutations, the cancer can grow based on this mutation. But if you completely delete P53, you get rid of this gene, the cancer cell can actually grow slower. So this particular mutated form can make cancer cell grow even faster. So you can see in some cases, a gene could be both oncogene or tumor suppressor. Okay, so it's you could use gene expression to help you guess, and sometimes you can also see some genes in one cancer type is mostly oncogene, but in another cancer type is mostly tumor suppressor, which will be kind of also interesting. So one thing that people found in terms of understanding the mutation rate is initially when people look at which mutations happen, they want to see for this particular gene, do you see a lot of mutations? And in the early studies, they found a lot of genes that I remember this lecture from Eric Lander. He's like, it's like a lot of smell and taste related genes, which is really weird because they are not even expressed in the cancer cells. How can they be recurrently mutating? So then people started to look at, this is one chromosome, say chromosome 14. If you look at the number of mutations per megabase, this is the red curve, which you can see across different locations on the chromosome, the mutation rate is different. And then they look at the expression levels of genes in those regions. You can see here, if the expression level is high, so it's opposite now. On the left side, when the high is high mutation, but on this side, the low level is here. This is bottom is high expression, top is low expression. You can see here, the regions that have high gene expression have overall less mutation, whereas the regions that have low gene expression, they have high mutation. Also, if you look at the DNA replication, you can see here, regions that have early DNA replication, they have low mutation, but regions that are replicated late have higher mutation. It's kind of interesting now. What we know is that this is looking at epigenetics. So regions that are replicated early on the chromatin, they are more open chromatin. Whereas the regions that replicate late, those are more heterochromatin. And we know that the open chromatin region overall have higher gene expression, a lot of transcription factor binding. They're kind of the more active regions in the genome. Whereas the late replication region of the cell has more heterochromatin, closed chromatin, lower gene expression, inactive chromatin. So can you guess why this is the case? Sam?
Have high gene expression that gene is more like
Yes, definitely one good reason is, so you can imagine, Sam said, if this region has a gene that's highly expressed in the cell, it might be important for the function. And so if you have a mutation, I would say mutation really randomly occur in the genome. If you create a mutation, most of the time you will make this gene work less well. Only in very, very rare occasions that you will mutate a gene that this gene will work better. It's like if you have a car, you randomly take things off from this car. You create mutations, like randomly take things off from the car. Most of the time, this car will not run as well. Only occasionally you take out some from the car will actually run faster, right? So most of the time you take out the gene that highly expressed, the cell will not work, it will not grow very well. Then over time it will be selected out, right? It will just grow at lower rate. Whereas if a mutation happens in those silent regions, no big deal. Nothing is happening there anyways, right? Then it's allowed to stay in there to become kind of a passenger. Another reason is regions that have high gene expression, if a mutation is created where the cell has truncation or frame shift, there is RNA nonsense-mediated decay. And so the cell actually can detect that, something is wrong with, so when the RNA is not making sense, there is transcription-coupled DNA repair. The cell can know that something might be wrong with my DNA. So it's gonna try to use the other copy, because we have a mom copy and a dad copy, it's gonna try to use the other copy to repair the potential DNA damage. And so those regions that have high gene expression, not only they have more essential genes, if something happens, the cell can also repair that DNA damage. Therefore, overall, you have less mutation. Whereas regions that have nothing going on, no gene expression, closed chromatin, mutation is allowed to propagate. This is also kind of related to gene length, because the genes in those silent chromatin regions are in general longer. So there is tumor evolution and selection, and also gene. I should add transcription RNA mediate, like expression mediated DNA damage repair. And so therefore, in order to really call recurrent mutations, we need to correct by the expected mutations in that region. So for example, remember in the early days when people look at which gene have recurrent mutation, they see these olfactory receptors, smell receptors, to have a lot of mutations. They initially call those as cancer driver genes, which is wrong. If they correct by the gene expression level or the DNA replication timing, you can see that the mutation level appears as expected. There's nothing more than what you expect. Therefore, that's not a driver gene. However, there's another gene in here based on the expression of the genes around this region and the DNA replication timing. We're expecting this level of mutation, but in reality, we're getting this level of mutation. That's a potential real driver gene. Okay, then you can use it to do this better. Yeah, so based on this reanalysis, you can now look at, like all the mutations that are recurrently happening in the tumor, adjusting for the expression and the level and the DNA replication level. And you can see here, the y-axis is pan-cancer. I think this is early day people looking at, I forgot, 9 or 12 cancer types. By this time, TCGA have 33 cancer types now. The x-axis is the recurrence of particular mutation just in the best cancer type. And you can see some of these driver genes recurrently mutate in not only one cancer, but also in all the cancers. A lot of the mutations are already known. There are some, like these red ones are novel, but there is a clear cancer association. But then there are some black ones that these are truly novel mutations that people detected. Most of these truly novel ones is from a point mutation. Traditionally, when people were looking at expression or copy number, they were not able to see it. But now, because we're doing whole exome sequencing, you really see that particular base repeatedly mutated, then you see them here, okay? Yeah, so really, have we sequenced enough? This is from a talk I heard from at AACR, it's American Association of Cancer Research. I was away last week because of this meeting. I think for 13,000 people were at this meeting, cancer researchers. And this is a talk I heard a few years ago. You think that, oh, we have already sequenced 10,000 patients, would that enough, right? So it really depends. If the mutation rate is at about 20%, meaning 20% of that cancer type patient will have this mutation, you probably only need to sequence 200 patients. By the way, Sam, do you mind helping me close the door because it's really loud outside? However, if the mutation rate is only 10%, meaning that only 10% of the patients with that cancer type have the mutation, then you probably need to sequence about 500 patients. Whereas if the mutation rate is only 5%, you just need to sequence deeper. Yeah, so there are some potentially weak driver mutations. Say only appearing in 1 or 2%, you will need to sequence even more. And this probably also tells us why in GWAS, if you have some rare variant, you have to sequence 100,000 individuals to really see it. Yeah, so most of the strong driver mutations have been found. Right now, the research is to understand, how these mutations, the function of these mutations, why they drive cancer. And the biggest surprise is the mutation on epigenetic regulators. In fact, over 50% of new and very strong cancer driver genes are on epigenetic regulators. There are some that are oncogenes. This is for DNA methylation. This is kind of related to DNA methylation. These ones are tumor suppressors when they are mutated. And some genes can be both, say oncogenes in solid tumor, but tumor suppressor in, say, lymphoma. And so recently there are also, it was a project that just started last year, is to expand the original TCGA. Most of the time, the TCGA samples are early, like stage 1, stage 2 tumors. you resect, big tumor out, you can do this profiling. And recently, this effort is being expanded to sequence the metastatic and drug-resistant tumors to really understand how tumor progress when they become resistant to the drugs, because the primary tumors don't kill patients. It's the metastatic drug-resistant tumors that really kill patients. But these type of tumor are much harder to sequence. Can you guess why? Yes. Yeah, so some students suggested maybe their mutation is too fast, so it's not representative. Sam? Yeah, or similar idea, maybe they are too heterogeneous. The answer is logistically it's hard. Primary tumor, when patients just have a primary tumor which hasn't been metastasized or only have local metastasized to the lymph node, you can resect the tumor and the patient will do fine. They will be cured. Once they are metastasized, say if you have a breast cancer metastasized to the lung, just taking out that breast tumor is not going to do much benefit to the patient. It doesn't help because you might know that it metastasized to the lung, but there might be other micrometastases to the liver, to the brain, or to the bone that we haven't seen. And so, in fact, over 100 years ago, people thought after surgery, if the patient still die, it's because they didn't clear, say that there's the radical mastectomy. They just keep cutting more and more out, right? Because they thought they didn't clear it enough. But the reality is because when the tumor has metastasized, you may not be able to see it. And so how much you cut just on the breast tissue doesn't matter anymore. And therefore, nowadays, we are doing much more conservative, just cut out the tumor if it's early. But if it's metastasized, we have no ethical reasons to cut them out from other tissue. Therefore, it might be difficult to get, right? For some tumors, it's easy. Say for head and neck, it's just outside. You just scrape it off, and there's actually no nerve. For melanoma, it's on the skin, it's easy. But say you have a liver cancer that's metastasized, you know the surgery is not going to benefit the patient. Why do we justify doing it? You could do brain cancer or other cancer metastasized to the brain, because even if the patient cannot benefit in terms of cancer, if the tumor is growing, the patient may not be able to stand, or it's pressing on something that controls their breathing or their other organ, balance or hearing or vision. You might want to take it out just so that they can still see or walk, right? Those you can take, but depends. It could be tricky, all right? Yeah. So.